Hello, my name is Dr. Bradley Alicea, and I'm with the Orthogonal Research and Education Laboratory. And on behalf of myself and the Representational Brains and Phenotypes group, I'm presenting on developmental neurosimulation, a route to embodied morphogenetic-inspired intelligence. So our starting point is the classical Braitenberg vehicle, and this was discussed in Valentino Braitenberg's book on Braitenberg vehicles in 1984. One of the ideas he introduces is the idea of this phenotype, and a phenotype-specific response to the environment. So you can see that he created these vehicles that have a specific phenotype. Most importantly, they have a sensor or a set of sensors and an effector or a set of effectors. In this case, an antenna is receiving light while the wheel is producing a movement in the environment. In addition, he proposed that there was some sort of hardwired network inside the vehicle that created this response. Depending on the topology of that hardwired network, there were different responses. He proposed that there was this first-order stimulus response mapping, and it does two things. The first is to observe the empirical world and then produces a hardwired response. The second is that vehicle types respond to things called taxis, or reflexive behavior. As you can see, there's this response to the light stimulus, and the vehicles either move toward the light or away from it. And so these are very simple neural networks that are embodied agents. Another starting point for what I'm going to show you today is this talk we did at NeuroMatch 2, and that was way back in 2021. And we did this talk approaching artificial intelligence as an embodied developmental process. So the idea is we can take inspiration from a number of systems where embryos are developing their shape, they're moving in some way, and they're responding to these same types of taxis. And so they exhibit maybe some of the same principles as a vehicle. One of the ways we've used Braitenberg vehicles as inspiration for this developmental approach is to model an age in embryogenesis. So at the top you see that there's a single circle with nothing inside of it, and that's supposed to be an egg. You see that there's progressive development from a three-layered circle to a circle with blobs, and then eventually you get this vehicle that we recognize from a few slides back. And so this is the type of thing that we're interested in, is how the shape comes to be. Can we shape new types of vehicles? And so age in embryogenesis is what we're calling this process, proceeds as a series of morphogenetic movement, or what we call morphological acquisitions, or a morphogenetic acquisition. And so you can see you start with a three-layered model in the image at the bottom. We start with a three-layered model that's undifferentiated. And then the black area migrates out into three blobs, and those represent the sensors and effectors. And then the outside ring actually inverts so that you have the beginnings of a nervous system. This is how, if we change the order of these around or we change the way that they proceed, we can end up with vastly different morphologies and maybe vastly different nervous systems. And so morphogenetic movements also allow for developmental computation. So if we think about morphological computation, we're doing developmental computation. And so we have these steps that we can follow. And in developmental biology, they have different stages of development where embryo actually reorders itself, sometimes pretty drastically, in terms of where cells are. Now, we're not dealing with individual cells here, but it's the same principle. Morphogenetic movements constrain what the adult vehicle looks like and how the adult connectome functions. So eventually we'll have an adult connectome. So we come up with this new concept called the Triploblast model of embodiment. We're dealing with embodiment, but we're dealing with it from sort of the developmental precursors of embodiment. We give the agent an initial complexity, and that's what we mean by Triploblast model. This is borrowed from evolutionary biology, where you have a three-layered embryo, or a triploblast. And we'll talk later about two-layered embryos, or diploblasts, as well. Each one of these layers have a specific function. Sometimes they can function together as they differentiate, and you can see that you start with a single layer, or a single undifferentiated sphere. You get your three layers, and then those three layers migrate in different ways to form the sensors, effectors, body, outer body, and then the inner chamber where the connectome is. Another concept we have are canalization functions. So we use these type of functions to model these different stages of development and how they're expressed. We also have something in tandem called community activation encoding. So community activation encoding is the way we're going to build our connectome inside of this embryogenetic agent. So community activation encoding is just simply a matrix of weights, or sometimes it can be a binary matrix. And so each developmental time point produces a connectivity matrix Wij and then each of these matrices have a corresponding determinant A. And you can map this to different types of phenotypes, so we can actually look at the developing connectome of C. elegans. And so we have an adult C. elegans here, a model of the adult, but we've highlighted the different neurons as they emerge at different time points. Neurons at different times, they're born, they maybe wire into a network, they become functional in some way, and we can determine those discrete time points by these different weight matrices and their corresponding determinants. So here we have the birth of neuronal cells in C. elegans embryogenesis at 265 minutes at the upper left, 280 minutes at the bottom, and 300 minutes at the upper right. As this network is gaining complexity, we can characterize this with these determinants. This is from a paper in Biosystems in 2018. Canalization functions are essentially logic gates that allow for developmental process to sort of proceed in different ways depending on the conditions that the agent finds itself in. So we have different logical in this canalization function that have to be solved at different stages of development. And depending on whether they're satisfied or not, they get on a different trajectory. So you can see this is a trajectory here where we have different logic gates kind of connected into this developmental tree. Our agents will follow these different pathways depending on their competencies at any given time. And so this allows for composition of behavior interacting with shape. So we can shape an agent's morphology, but we can also kind of shape their connectome at the same time and thus shape the behavior of the adult. We can put this into an artificial genome. This is a circular genome map where we see that there are different points on this genome map that correspond roughly to different points in the canalization function. So this is a series of instructions that go in a certain direction along the genome. As those instructions are read out, they hit these canalization points. So you can see examples here where you start out with an undifferentiated agent. At the lower right, you have a switch between a triploblastic and a diploblastic form. At another point, you have a switch between the effectors being on either side or having a tail, and later lead to more distinct phenotypes and more specialized phenotypes. So this is a tree showing a single undifferentiated agent, and then you have the triploblastic lineage and the diploblastic lineage. So in the diploblastic case, we can take those two layers and we can create a swimmer. In the triploblastic case, we can use a 3-layered architecture to create a roller. And so you can see the roller can lead to more classic-looking Bradenburg vehicles, whereas the swimmer leads to either non-functional adult phenotypes or these swimmers. See, the idea is to get a diversity of shapes through development and a diversity of nervous systems, and they'll exhibit different types of behaviors. So now we move on to the morphogenesis of the connectome layer. And what we do here is we're using Alan Turing's undifferentiated machines. And basically, there are two stages here. We have a morphogenetic stage and a learning stage. So the morphogenetic stage is where there's an increase within the connectome itself of unorganized machines, or very generic types of finite state machines. Those are A-type machines. And they form modules, which are these Boolean finite state machines. And then we have this learning stage. So this is where we have stability in the network. We have enough A-type machines in place, but then they begin to get replaced with what they call B-type machines, which have a different morphology in them. And so this is where we get this developmental freedom or learning in the connectome layer. So we get this sort of morphogenetic stage where we grow the network, and then after we grow the network, we replace it with more specific parts that are specialized to different behaviors. This is where the network can exhibit developmental freedom or this learning, and it can specialize to different tasks and things like that. So developmental freedom is the ability to learn on a stable network. And that's not simply changing the weights or optimizing the weights. It's actually changing different aspects of the topology in a very specialized manner with different things that can represent semantics or represent other types of feedback loops that are localized. Another aspect of classical Bradenburg vehicles that we can draw from is this idea of having these behaviors that aren't just taxis. They could be almost mirroring emotions or emotional states. So you have these rudimentary behaviors that Bradenburg observed that are characterized by Bradenburg as love, aggression, or happiness. They're kind of anthropomorphic in a sense because he didn't really make rigorous tests of whether they were these behaviors or not. He just called them these behaviors. So love would be like being really attracted to light. Aggression might be being really attracted to hurting your neighboring vehicle, where happiness might be just going around in a circle, these sorts of things. So it's not really based on a formal representation, but that begs the question then, how do we achieve richer, more robust symbolic behaviors? And so we've done a bit of work here on looking at behavioral training and cumulative culture on gradients. So cumulative culture is this idea where you have a bunch of vehicles and they exhibit collective behaviors around a common resource point that forms a gradient. So we have gradients of resources, gradients of different things in the environment, different stimuli, and we talk more about this in this artificial life paper from 2022. And so this is an example of a gradient, and I'll show you, this is from Ankit Gupta, who's one of our co-authors. He developed this technique called BV collectives, or Bradenburg vehicle collectives. And as you can see, there's this 3A friendly stimulus, and you can see movement of the agents around that common stimulus, and they have a certain sort of individual behavior, but they also have collective behaviors around that. Now, if you take two 3A type behavioral anchors, so this 3A here in this movie on the left is a behavioral anchor. On the right, we have two behavioral anchors. So what will happen, it's kind of like the sun in the previous examples where that's their core stimulus, and then they behave around that. So if you can see the movie, the agents have a different behavior around that. They group in sort of two clusters, and then their agents moving around those two clusters as opposed to one cluster. And so if we recall our canalization function example where we had our tree of developmental outcomes, this allows again for the composition of behavior and shape. And so that leads us to other types of work that we can do with shaping the sensory inputs. And so this is where we have an example of sort of an antenna that you can have on the front of the vehicle where you have multiple inputs and they get summed going into a canalization function and being processed in the connectome. So we can do a lot of pre-processing of environmental information in a way that's very reminiscent of dendritic computing. We can also play around with the surface. So if you don't like to have a flat surface or a regular surface, you can have an irregular surface. And of course, the irregular surface, as you might imagine, responds to the stimulus in a different way. So we can not only have these collective behaviors, but we can have these sort of different types of pre-processing, different types of morphological computation going on as well. Finally, we have some inspiration from a book that's coming out by Jeff Yoshimi and Scott Houghton. It's on Bradenburg vehicles, but they've been talking about this on social media with some of the things that they've observed. Scott Houghton was discussing something called paratrichoids. And so these are geometric shapes that have different circles that orbit each other. What my point here is that they produce these outputs that are constrained by the geometry alone. So you don't really need a connectome to do anything. They have these different dynamics, or very interesting dynamics, based on their interlocking geometry. For an epitrichoid, there are 4 rolling discs, each with an equilateral triangle attached to its interior. So you can see that, and they move accordingly. So there are all sorts of interesting connections and behaviors you could make to that. There are a lot of dynamics to explore there. And then the reason I bring that up is because it has a parallel to something we found in this artificial life paper, which is that, there's some interesting things going on in our vehicles that we had, and developmental Bradenburg vehicles, that resemble what they found with paratrichoids. And so this is maybe an example of geometric complexity as some sort of behavioral constraint. So again, we're taking this sort of eco-devo approach to neurosimulation. Ultimately want to look at different timescales from single trial learning to different types of internal mechanisms, to long-term memory, to the lifespan of a vehicle, to historical memory amongst vehicles, and then finally the evolution of vehicles over many generations. And so we hope to look at the effects of that, as well as the sort of the development of vehicle morphology in the future. So join us for Google Summer of Code. You have an opportunity to participate in the Representational Brains and Phenotypes group. We're sponsored by INCF, so check out their GSOC board if you're interested in participating. And thank you for your attention.